Hello, everyone, and welcome to today's webinar, CiPA Initiative and Validation of High-Throughput Methods to Characterize Compound Effects in Human iPSC-derived Cardiomyocytes. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Molecular Devices and Eurofins Discovery. To learn more, please visit moleculardevices.com and eurofinsdiscoveryservices.com. Now, before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or let us know you're experiencing a problem by using that Ask a Question box. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I now present today's speakers. Dr. Rengi Ranganathan, Senior Scientist at Eurofins Discovery, Dr. Panita Thurkiatmangal, Senior Scientist, Group Leader of Cell-Based Screening at Eurofins Discovery, Dr. Oksana Sharenko, Senior Research Scientist at Molecular Devices, and Ms. Carol Crittenden, Application Scientist at Molecular Devices. For complete biographies on our speakers, please visit the Biography tab at the top of your screen. Our agenda for today will include Rengi discussing the importance and challenges of cardiac safety evaluation and how the CiPA initiative offers new ways of testing potential compounds. Then Panita will discuss ways to analyze calcium flux data from cardiotoxicity studies from the CiPA 28 compounds. Oksana will follow that up with additional data from the new Flipper Penta assays. And finally, Carol will discuss the new features of the Flipper Penta system, including high-speed camera, which enables more detailed cardiomyocyte and neuronal data, as well as peak pro analysis options that were used for data analysis throughout this presentation. And finally, we will conclude with our Q&A session. Our speakers will now begin their presentations.
Hi, my name is Rengi Ranganathan. First of all, on behalf of Eurofins, I would like to extend a warm welcome for this webinar, wherever you are. And I'm going to, I have about 20 years experience in the cardiac safety evaluation. So I'll share with you some of the, the challenges, how the recent technology is pushing the boundary to make the drug discovery a little bit more efficacious. So the outline for my talk is going to be first, we are going to set the tone for understanding the rest of the talk by describing, what is it cardiac depolarizing current or plateau phase or repolarizing currents of a cardiac accident potential, how it is connected to the QT interval, and because these are the basic parameters that are going to be measured even in the preclinical or in clinical drug discovery process. So, and then I will briefly talk about the drug-induced QT prolongation and Torsades du Pont, one of the parameters for this deadly disease. And the current safety paradigm outlined in ICS S7B and E14, I'm talking about from 2000 to 2014. These were the guidelines used for drug discovery. And there were some issues in there, which is summarized in this point. That is the surrogate endpoints, human ether-go-go related gene block, and QT prolongation was used as a surrogate endpoints, not the proarrhythmia risk, the Torsades risk, because the Torsades is very rare to see in clinical trials. So they have to look for surrogate endpoints, and that's how they started using HERG block and QT prolongation. The issue here is the HERG block and QT prolongation is very sensitive, but it's not very specific for predicting proarrhythmia risk. And I also want to talk a little bit that the Torsades starts with the QT prolongation, but not all QT prolongation ends in Torsades. So from the get-go of these guidelines, we know the decks were against the drug discovery. And, over the 15 years, they figured, the consortium have came up together and tried to push the initiative based on some experimental evidence, which I will briefly talk about in the rest of my talk. The initiative is to facilitate the adoption of a new paradigm for assessment of clinical potential of TRSAD, not measured exclusively by a HERG block or not at all by QT prolongation. And then I'm going to step in a little bit about how Eurofins joined the CiPA initiative and contributed, for the high-throughput platform by scanning the 28 drugs that, span high, intermediate, and low TRSAD risk. And then we separately tried the multi-electrode array using iPSC-induced pluripotent stem cell, of course, human cardiomyocytes in a multi-electrode array with specific blockers for, some of the depolarizing and repolarizing currents, which I will share it with you. And then comes my colleague from San Diego will talk about how she used the flipper as a tool to help discover to help the guidelines. All right, the next slide. Here, I'm going to just summarize it in simple terms. So cardiac membranes are very highly polarized. What do I mean by that? If you look at the cardiac cell, the inside of the cell is very negative, close to minus 80 millivolts, compared to the outside of the cell, which is close to 0. And and also, if you look at the concentration gradient for calcium and sodium, they are much higher on the outside of the cell than the inside. And the voltage gated channels are, membrane gated, and they are poor on the membrane. So when they open, the concentration gradient, depending on the concentration gradient, the sodium and calcium will, in this case, will flow into the cell. And because it's the reverse for potassium, when the potassium channel opens, it flows out of the cell. Why did I put this diagram? Because in the minus 80 millivolts, when the sodium and the calcium opens, these positive ions go in and then neutralize the negative charge, and they would become more positive. Whereas when the potassium channel opens, the positive channels will go out. Then they try to bring the resting potential back to the minus 80 resting membrane potential, and that's what I'm going to try to show here in this cartoon. So the first is the depolarizing currents. The currents here is the black line is the baseline, and the blue current is the bottom of the baseline. That means it is flowing into the cell. So these positive sodium and calcium charges flow in, neutralize the minus 80 millivolts, and that's what you see here in this left-hand panel, that minus 80. Once the sodium channel opens, it goes to plus 40, and then comes the calcium. That's where the plateau phase is, where the inward and outward, the depolarizing and repolarizing current are more or less similar, same amplitude, so it's a kind of plateau phase. And by the time the repolarizing current comes, both the sodium and calcium contribution is over, they are inactivated, they will not open unless they come back to the minus 80, and then they come back, the cycle continues again. So then comes the rest of the potassium channels, and for that particular importance is the IKR. This is what the HERG current is called. This is a beautiful current. The way it is set up in nature is amazing to see that it opens only at the repolarizing stage, and the maximum contribution comes at that stage. And that's why it is very important for the repolarizing phase. So now we have established the depolarizing, what is depolarizing means, what is the plateau phase means, what is the repolarizing current means. We can go on to the next level, and I will add one simple thing for people, that depolarizing current is the one which triggers the cardiac muscle contraction. And then the repolarizing current is the one which relaxes the muscle. And that's how one cycle of cardiac beat happens, just in case, if you don't know about that. So this slide, if you record the cardiac action potential from different parts of the cardiac tissue, this is color-coded to present and show the propagation of the cardiac electric signal is from the top to the bottom. And so this starts, the blue cardiac action potential starts first, and at the end side of the bottom with the ventricular muscle. So if you align them and summate them, and that's what you get as a normal ECG. And this is given in the Atlas of Human Physiology, page 17, if you want to read more about it. So the P wave comes from the atrium, and the QRS comes from the ventricular depolarization. And remember, the left ventricle is the most powerful muscle, and it pumps the blood for the rest of the body. So the QRS and the T is the repolarization. So the QT interval is the one which measures the left ventricular function, particularly the pumping of blood to all over the body. So by establishing this, we're going to go to straight to the problem which happened in the early part of, or the late part of the 1990s, 1998 and 2000. That's the time that the drug-induced QT prolongation was observed, and they also figured it out. It was the HERG which was doing it, and the threshold frequencies were far too low to be detected in clinical trials, so it was only found in phase 4. After the drugs were approved, it was out in the open market, and then they saw the threshold, then they had to withdraw. So within the 2 years, they had to withdraw 5 compounds: pepfidine, cetindole, esomeprazole, revefloxacin, and cisapride. Now, let's give some thoughts to the pharma industry. So they spent 10 years developing each drug candidate and about $1 billion, and suddenly they had to take it back, and 5 drugs had to be withdrawn. So the whole pharma industry was shaken up, and then all the pharma industry, FDA, and other guys got together, and they formed the other initiative that's called the S7D in 2000. So what did we learn from these all 5 drugs? They are all potent HERG blocker, very highly selective in the pharmacodynamic range. What I mean that is when you take the drug for a disease, it's the same concentration. It not only cures your drug, it also blocks the HERG. So you don't want to die by blocking the HERG, and that's why they withdraw the drug. So, and then they also knew that the regulators knew there is no clear markers for these problems. They suggested to use HERG and QT, and this was introduced in 2000. And if you go back to the next slide, how HERG induced the QT prolongation. As we talked about, HERG is the human lethal-go-go related gene, a funny name. It's a potassium current. The IKR stands for I for current, K for potassium, R for the repolarization of the cardiac action potential. This channel is very unique. It has the inside core is very unique in that sense. It promiscuously binds structurally different compounds. So it is a very 30% of the drugs have some HERG block. So there was a very big problem for the drug pharma industry. This inhibition, what happens is, as we talked about in the action potential cartoon, when the HERG channel the current is blocked, the repolarization takes more time to do it. So there is a delay in repolarization, and that can reflect in QT prolongation, and that's okay. But it's only when you see this early after depolarization, when there is a prolongation, and at that time, any changes in the inverted output current, because of the plateau phase, the resistance is very high than the rest of any phases of the action potential. Any small changes in the current at the plateau phase can induce EAD. Most notably, the calcium current or the sodium late current, which I forgot to mention in the last slide. So this EAD is very rare, and that's the one which triggers the mechanism for threshold. So what I'm setting up for the rest of the talk is HERG block is okay, QT prolong is okay, but is the threshold or EAD, that's not okay. So if you are blocking HERG, looking for HERG block and QT prolongation, you are already failing most of the drug candidates, right? So that is the issue for the pharma. And we can't blame pharma because they spend a lot of money and time. So what they did was, the HERG became known about the pharmacology known about 1996. They stopped any drug that blocked HERG at a particular range. They had some kind of a safety ratio, and if that safety ratio is crossed, they stopped the development for the drug. So when they used that approach, in the 1996, there was 5 drugs withdrawn in the 5-year time period from 2005. In every 5 interval, you can see the number of compounds getting withdrawn is coming low and low, and from 2011 onwards, there was no drug withdrawn because of the threshold. It was a very good success, but at what cost? And that's what we are going to see here in the next slide, because the QT prolongation is not always leads to proarrhythmia, and we also know HERG inhibition leads to QT prolongation, but it's not always equal to proarrhythmia. So, and we have some specific cases of drugs too, like Prozac, a multi-billion-dollar drug, is an antidepressant. It blocks HERG potently, but at the same concentration blocks calcium too. Similarly, the verapamil too. The ranolazine is another case where it blocks HERG as well as the late sodium current. The calcium current, the lead sodium current are the players at the plateau phase, and that's why blocking these two currents, they are very cardiac safe, in addition to blocking HERG and inducing QT prolongation. And that's what is shown here at the bottom panel figure. The left panel is for only HERG block drugs only. So for the carbon gives it three channels, sodium, calcium, and HERG. And the sodium and calcium not blocked, but if the HERG is blocked, when you block the HERG, you have the prolongation of the action potential duration. And because the calcium, the lead current could be active, it can induce, not all the time, but certain time when the conditions are very ideal, these currents can induce the EAD, and that can lead into the preferred pond, which is very detrimental for the people. Now, let's take a look at the balanced ion channel block, where a drug, a single drug when you apply, it not only blocks HERG, it also blocks calcium. It may also block the lead sodium current. Let's see what happens here. Yes, you do see a prolongation in action potential duration. Yes, of course, you see the QT prolongation, but you never will see any preferred. So we knew about this in the early 2000 or 2005. So that's what triggered for the new initiative, and it took a while for all of us to come together in 2011 or 12. And they proposed this comprehensive in vitro proarrhythmia assay, CiPA for simple. So it has four panels, listed here. The first is to look at multiple ionic currents, drug effects on in vitro assessment of drug effects on multiple ionic currents. And all these effects are integrated into in silico computer modeling to predict proarrhythmia risk. All right, this model has been validated. And so they will, once the arrhythmia risk is validated, then they will look for unwanted or unanticipated effect on in vitro stem cell ventricular cardiomyocytes from human. And that data will help to determine whether there is any effects or not. Then you go for the phase 1 clinical trial. And this here, what they are looking for is, again, they are going to look for exposure response analysis. Most of the in vitro people will know concentration response, whereas in the animal, in vivo animal, that they use different kinds of technology, but they now come to the conclusion that exposure response, that is, for any time point when they measure the effect, they will know what is the concentration of the drug at plasma. So that way they can figure out what's happening for the QT interval. Even in the QT interval, they're not only measuring the QT interval, they are measuring this, in this publication, if you have more interest, you can go look at it, that from this point to this point is called the J to T peak, and then the T peak to Tn. The drugs that block the calcium and the sodium current, they do not have any effect on the J to T peak, or also called the early repolarization phase. But the HERG-only block will have an effect on the T peak to the Tn. So they have adding more parameters to help discover the drug discovery more efficient way. So what we're going to summarize on this particular slide, take-home message is, if successful, this initiative is successful, CiPA will enable, create a pathway for drugs with the HERG block and QT prolongation to advance without intensive ECG monitoring in phase 3 trials. That's called the thorough QT trial. That will take a lot of time and money, so you don't have to do that anymore if CiPA passes through. And also the second important factor is that you can enable updating drug labels to be more informative about proarrhythmic risk rather than a general QT prolongation. This is all helping the pharma industry and also bringing more drugs into the human population. Now, in this slide, I'm going to explain a little bit how we often join the CSRC, Cardiac Safety Research Consortium, and the HEC and the FDA high-throughput screen subteam in 2015. What's the idea on this is, different people, different groups, different platforms, but you use blinded compounds shipped from one source, and with you will all use standardized assay protocols to collect data in their respective platform, and then send it to the FDA, and they will look at the data for, how good is this reproducibility across the different sites and platforms. And here we let, some of the compounds which have very low risk, and intermediate preferred risk, and high preferred risk. I'm not going to go to all the data here. We have sent all the data to the FDA, and which is on our website about all this, the concentration response and everything. What I'm showing here is that we put a premium on our vehicle control data. That is, why do we do that? Because we match the time match the vehicle control to the compound addition to show that if you add only vehicle, there is no effect on the baseline. But if you add the drug at the same time concentration, you have a clean dose response curve without any effect on the baseline. And so you have a much confidence in your IC50 value estimation. Here in this case, it is on downstream citron, as IC50 is close to 1 micromolar, and this is very close to what we have seen with the rest of the groups too. And similarly for the sodium 1.5, which is specific for the cardiac sodium channel, I'm sorry. And here it's a peak current is measured, and I'm showing it peak current. I didn't want to put a lead current, in the slides to overwhelm you with the slide with a lot of data. What it shows here again is that it's a stable baseline at clean dose response, and you can take the IC50 value very clearly without any doubt. For coming to calcium, we got some issues with using CiPA protocol. It had a tremendous rundown, more than 40 to 50% rundown, which is not a good QC for our assays. So we had to tweak the protocol and use a different way to bring the rundown to minimal, like 15 or 20%. So that's why this is not a CiPA protocol, and there's a specific reason why we did it. And we also used the specific blockers, nifedipine, metindipine, to show the reproducibility is very close to each other with the minimal rundown. And so the work on the HERG results in 2 manuscripts in the works with the FDA. I'm not going to read this, but it's in there if you want to look at it, and if it's going to be published in the near future. Hopefully, you'll get more detail on this. And so one aspect of the CiPA is using induced pluripotent stem cell-derived cardiomyocytes, obviously from human, as an in vitro model to study cardiotoxicity, of CiPA compounds. For that, we use the MEA, that's the bottom signal, and Panida used the Flipper cardiac calcium signal assay. So we'll talk more about it in the next few slides. This is the master platform listed here on the top, and this is the cartoon here compares what you see to the ECG. The Q2 interval is to do the field potential duration. And this spike, sodium spike, is the cardiac depolarization signal, and the T wave is the cardiac repolarization signal coming from cardiomyocytes. And the period between the depolarization and repolarization between sodium spike and T wave is the plateau phase, mainly coming from calcium channel. And this is very important. Why the 4 salient features is that it is done at physiological temperature, as it's in the body temperature. It's an integrated measurement of all the ion channel data, and you can translate that, and you can check for anticipated or unwanted effects. So here again, the three critical phases of the cardiac safety: cardiac depolarization, plateau phase, and repolarization. We all know coming from sodium, calcium, and HERG channel or potassium channels. So if you use a specific mexiledine blocker, which is very specific for sodium channel, it decreases the amplitude for cardiac depolarization, as one would expect, without doing anything to the rest of the two, the plateau phase or cardiac repolarization. On the other hand, when you use the nifedipine, which is very specific for calcium channel, it shortens the duration between the depolarization and repolarization, and that's what the arrow marks. The T waves start moving to the left, close to the depolarization. It means field potential duration becomes shortened. It's the reverse for the HERG blocker E4031. It prolongs the repolarization wave, and it becomes longer and longer. Sometimes you can even see early after depolarization in these wells. So without much detail, I'm going to just summarize here what I have seen in the MEA parameters. The key 3 parameters are, the beat period, if any effect on the beat period, and in the field potential duration and spike amplitude. The beat period picks the calcium, calcium blockers. Field potential duration, again, you can see both the calcium and HERG blockers too. So let's go through quickly. The E4031 is a HERG blocker. It prolongs the beat period and the field potential duration with not much effect on spike amplitude. The DC stands for Fred reset correction, which is for beat period. If any change in the beat period can be corrected, so comparing apples to apples in one way. And the mexiletine is a sodium channel blocker, did not do anything for either the three of those, but only had an effect on spike amplitude, which is the depolarization peak. And nifedipine diltiazem cardiac calcium channel blocker, the beat period became decreased. That means that it had more beats per period. And you can also see that reduced the duration in both cases. It was not much effect on spike amplitude. On the other hand, with the vehicle control that matched the duration of these compounds, Edison is given as a control to show what kind of effect you would see. And that's it from my side of the talk, and I'm now going to hand it over to my colleague from San Diego, Panida. Panida, please take it away.
Yeah, thank you, Ranji. So for the next part, I will be presenting the application of iPSC-derived cardiomyocytes as a model to evaluate potential cardiotoxicity of compounds using FLIPR. For this study, we use calcium flux assay to monitor contraction and relaxation of cardiomyocytes. We detect either increased or decreased levels of calcium ions using calcium-sensitive dye. The dye binds calcium ions in cytoplasm as displayed in the cartoon on the left and elicits fluorescent signals that can be captured by a high-speed CCD camera in FLIPR. So the detected fluorescent signals from the calcium flux assays represent intracellular calcium oscillations. In order to understand what happens to cardiomyocytes upon compound treatment, I use PeakPro, shown here on the top left panel, which is an add-on of ScreenWorks to extract kinetic parameters from the traces. PeakPro assigns peaks, which are displayed as blue dots. It can also set the baseline and the triggered levels as shown in the red and green lines. The increase in fluorescent signal corresponds to contraction, whereas the decrease corresponds to relaxation of cardiomyocytes. We can monitor in real time and over time. In this example, the second panel on the left is the pre-treatment trace. Then the cells were treated with compounds and incubated for 30 minutes, and then we read, shown in the third panel on the left. There are several parameters that we can extract from the traces using PeakPro. From our experience, mean peak rate, calcium transient duration 90 or CTD 90, and peak spacing are pretty consistent across the whole cell plate and from experiment to experiment. So we go with these parameters to plot our dose response curves as shown on the right to calculate IC50. These are more examples of cardiomyocytes treated with isoproterenol, which is an agonist of beta-adrenergic receptor that can increase the heart rate. The trace exhibits increased mean peak rate and decreased CTD 90 and peak spacing. These parameters correlate with the effect of isoproterenol, as you can see. The next one is estamazole, which is categorized in intermediate risk of TDP. Estamazole is an ERG blocker and causes QT prolongation. From the trace, the mean peak rate is decreased, whereas CTD 90 and peak spacing are increased, which are characteristics of peak prolongation due to blockage of repolarization by ERG. The next one is sotalol, which is categorized in the high risk for TDP. Sotalol is a beta blocker. It can also prolong repolarization by blocking the rapid component of delayed rectifier potassium current. From our assay, sotalol-treated cells exhibit decreased mean peak rate, but increased CTD 90 and peak spacing, showing the characteristic of peak prolongation as well. For these three parameters, our data points are quite tight, as shown in these three graphs. And the blue dots, they are the 30 minutes post-treatment that you can compare to the pre-treatment. So you can clearly see these data points, they are clearly distinguishable between before and after the drug treatment. And these scatter plots demonstrate that our calcium flux assays is uniform and consistent. The observed effects are also correlated with actions of the drugs. More assessments of CiPA compounds will be discussed by Oksana.
Hello, thank you so much, Panida. So in the beginning of my part of presentation, I would like to show you a video. So on this video, you can see those flashes up and down. So they occur when cells contract synchronously and calcium goes in and out of these cells, intracellular cell storages, and calcium dye get brighter or duller accordingly to concentration of calcium in a cell. So you can see control recording, and also you can see changes in a pattern for cells treated with epinephrine or verapamil. So with epinephrine, you can see increase of amplitude and frequency. And with verapamil, you can see decrease of amplitude. So as shown on a video, so I would like to describe what is our assay is doing. So on that slide, you can see that we actually absorbing cells which are plated in a culture, and those cells are cardiomyocytes, iPSC-derived cardiomyocytes, and you can see them in 10x magnification. And when you put a dye on it, you can observe those fluxes up and down, which you can measure with our instrument, Flipper Penta, and also you can record and characterize those peaks. So for cardiotoxicity evaluation of 28 CiPA compounds, we have taken measurements of calcium oscillations, and it seems like a promising method for toxicity assessment. We evaluated a set of CiPA compounds which categorized as a high, medium, or low risk categories according to clinical data. We have used iCell cardiomyocytes 2 from CDI, which were cultured as recommended for 6 days, and then loaded with early tox calcium dye and treated with compounds. So the recording could be done at different time points. So starting from 15 minutes after compound addition, but also additional recording could be taken at 30, 60, 90, up to 24 hours. Also, you can do long-term compound treatment. In this case, you treat with compounds first and then add the dye to the cells and record the effects of those compounds with Flipper Penta system. So in this slide, you can see a screenshot from a Flipper instrument. So here, we've tested high-risk compounds. Those compounds were tested in quadruplicates, and you can see the effect of compounds increase to the top of the plate, where it was increasing concentration. So you can see also some negative controls, which amoxicillin and biotin on the right side of a panel, where you can see no impact on a pattern, but many compounds which considered high risk show very severe changes in a pattern. So the most notably, you can see prolongation of the peaks, and also you can see some secondary peaks appearance. In addition to that, you see changes in amplitude, beating frequency, also regularity, up to complete stop of beating rate. So here we can present individual patterns. So on this slide, you can see in blue the control pattern, and in red the pattern affected with indicated compounds. So you can see those different patterns, modifications, which I just described. So you can see prolongation of peaks with astemizole. You can see increase or decrease of amplitude Like with isoproterenol, you can see increase of amplitude and frequency. With others, you may see decrease of amplitude and frequency. You can see increase in between peaks, also irregularity, and you can see stop of oscillation in some concentrations. So those typical oscillation patterns could be measured and characterized. So our software is able to detect main peaks and also secondary peaks. It can also measure the amplitude, frequency, rise and decay time of peaks. And it can characterize distance between peaks and also regularity or irregularity patterns. So here you can see some additional patterns where you can see combination of different effects. For example, you can see irregularity and appearance of additional peaks. Also, you can see this pattern which we call like stop oscillation, where you can see decreasing amplitude and also highly increased frequency, which actually cause stop of oscillation. So with our software, you can characterize those peaks and derive number of measurements. Those descriptors include peak count, peak frequency. You can count main peaks and also secondary peaks, and number of secondary peaks per main peaks. You can count distance between peaks, amplitude, and also rise and decay time. So this number of descriptors can characterize all complexity of patterns you can observe. So some of those readouts have a good dose dependence pattern. Like for example, on this slide, you can see monotonous decrease or increase of peak frequency. Also, you can observe monotonous increase or decrease of amplitude, but some other readouts may be non-monotonous. So for monotonous descriptor, you can calculate EC50s. For non-monotonous, you can actually calculate minimum concentration at which the change occurs. So in this example, you can see high risk, medium risk, and low risk compound and changes in peak count over concentration. So notably, high risk and medium risk compound cause mostly decrease on beating rate, and low risk compound, many of those actually cause increased beating rate. So you can characterize different measurements and calculate effective concentrations for different measurements, and you can use multi-parametric approach for toxicity assessment. So interestingly, we try to look at these concentration dependencies the way that we would normalize it to Cmax, so the actual concentration of compound in a blood. And we also look at different readouts, actually effective concentrations or EC50s for different readouts. And what was interesting is that for high-risk compound, we observe that those changes, especially increase of peak prolongation or appearance of secondary peaks, appear at concentration which are comparable for Cmax. For medium-risk compounds, you can see those changes appear at concentration about 10-fold higher than Cmax. And for low-risk compounds, you don't see any of those effects at all, like increase of peak prolongation or appearance of secondary peaks, while you still can see stop beating or increased fibrillation or irregularity or change in amplitude at different concentrations. So that would suggest that peak prolongation and also appearance of secondary peaks may be the most important parameters for prediction of potential Torsades de Pointes. In addition to characterization of oscillation pattern, we also follow with high-content imaging to characterize changes or in cell viability and cytotoxicity effects. For example, out of those 28 compounds, we had several which had cytotoxic effects. So this is kind of important to see whether changes caused by compound are due to cytotoxicity or changes in oscillation pattern. So we use image express instrument for this purpose, but any imaging instrument could be used. So, and we characterize the number of live and dead cells and also cell spreading and intensity for viability markers. So in a conclusion, seems like the assay is very sensitive for detection of various compound effects on cardiomyocytes, and these effects can be measured and evaluated by peak pro software. And most notably, EED occurrence and peak prolongation, apparently the most distinctive and predictable measurements for Torsades points. So now I will transfer to Carol, who can tell more about the instrument and software.
So this is Carol, and I'm going to be talking about using the Flipper Penta system for cardiotoxicity assessment, as well as some of the other things that the Flipper can do, and we kind of look at it as two more additions to the family of things that can be done with Flipper. So the Flipper Penta system has an unmatched flexibility. It's been around for more than 20 years, and it has grown as the technology and industry has grown as well. And it's flexible because you can change heads, you can change LEDs and emission filters for different wavelengths. You can have a format of assay flexibility if you want to use a cell suspension system. It has a sensitivity flexibility with the cameras. We offer two main cameras. One is a standard EMCCD camera, and then our newest Flipper Penta option is the field upgradable, and it's also a 100 Hertz option that still has luminescence capability. And using this camera, it's able to do peak pro calcium oscillation measurements, and then also with automation, it works beautifully. And the nice thing about Flipper is that you can start, if you're in a 96-well format, you can grow as your assays grow and add things in the field as well. The newest features of the Flipper Penta system is the high-speed camera, as well as the ScreenWorks Peak Pro 2 software module, which is good for analysis of calcium oscillation peak characteristics. And it has a difference in temporal resolution. So most of the standard cameras would give you analysis that looks like this, and this is at 8 images taken per second, or 8 Hertz. And you don't necessarily get enough detail in the peak to see what's going on. And this is the same well, same cells, at 30 Hertz using the new camera. And as you can see, it's easy to identify both the peaks as well as the secondary events involved. Flipper Penta system is able to do the assays of your choice. For small molecule screening, it's the standard for GPCR assays, any other calcium ligand-gated ion channels, other calcium assays. We have a FURA2 assay. It can do a Corin assays, which is a luminescent assay. We can do membrane potential assays. We have a potassium assay. And also for toxicology screening, which we've been talking about here today, we have the cardiomyocyte toxicity assay, measuring calcium oscillations in cardiomyocytes, as well as a neuronal toxicity assay as well. We didn't talk about that here, but we have other information about that. And more. We can do bread assays, we can do all kinds of other dye assays, and you can look at oscillation in spheroids as well. So the calcium oscillation analysis in Peak Pro 2 gives you a number of parameters that you can look at to characterize your peaks. We still are carrying the Peak Pro 1 module. It's available, and then we have the Peak Pro 2. And Oksana gave a good description of all of the different characteristics of the calcium oscillation peaks to be able to identify with ease. The ScreenWorks screen basically looks like this. You can look at the detail screen. You've got your selected compounds that you're looking at, the control screen to identify the wells and parameters and controls of the peak analysis, as well as a chart for looking at all the different parameters, which is easily exportable. And then you can form your graphs. And shown here are 3 different characteristics for one of the higher CiPA 28 risk compounds, astemazole. And you're looking at the change in peak rate with concentration, as well as peak amplitude, as well as the mean peak decay time from 90% to 10%. So that's easily done. And the benefits of the Flipper Penta system, which is our newest offering, is we have the 100 hertz camera, which enables new biology and a better understanding of both neurons and cardiomyocytes, easy to switch to luminescence, and easy to switch to all the regular assays that the Flipper has always done. And then we have the greatly enhanced peak characterization of calcium assays with the Peak Pro 2 software. So I'm just going to give a quick summary. Renji gave us the good perspective on the importance of drug safety and CiPA 28 and methods of detection and analysis, and the correlation of calcium oscillation, EFIS, MEA, and the clinical Cmax was discussed by both Oksana and Renji, and iPSC cardiomyocyte calcium oscillation measurements using the Flipper Penta system and the ScreenWorks Peak Pro 2 software. So I want to thank you on behalf of the 4 speakers, and we're going to turn it over for question and answers.
Thank you, Carol, and thank you to all our speakers for their informative presentations. We will now start the live Q&A portion of our webinar. Now, if you have a question you would like to ask our speakers, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many questions as we have time for. Okay, now let's get started. Oksana, let's start with you. Do you need viable cardiomyocyte cells for your assay? And if so, how do you obtain them?
So this is pretty easy question because you can derive your own iPSC-derived cardiomyocytes, but we have taken advantage of companies who actually do it professionally. So we collaborated the most and work with CDI, Fujifilm CDI company. So we get cells from them, and there are also other vendors like Encardia or Stemonics where you can get some models as well.
Thank you. Now, Renji, let's come over to you for this next question. Have you tested the 28 CiPA reference drugs in the MEA platform? And if yes, what was the predictivity?
Yes. The CiPA 28 has been done, and even like 3 or 4 years back, there's a few publications came out too on the sensitivity of this MEA assay. So we have what we have done is we focused on only on the parameters, which is very pertinent to the cardiac safety. For example, the cardiac depolarization and repolarization and plateau phase, and we've been focused on that inhibitors first, but we've been collecting data on these 28. Just to understand how the system works. So it is in the works.
Thank you. Now, Carol, this question will be for you. What is the highest frame rate for the Flipper CCD camera?
So the highest frame rate for that high-speed camera on the Flipper Penta is 100 hertz or 100 images per second.
Thank you. Renji, let's pop back over to you. This next question is for you. It was mentioned in the beginning of the presentation that HERG and QT prolongation were very sensitive but not very specific. Do cardiomyocytes give us the boost in specificity that HERG and QT lack?
Yes, this is a good question, but that's the bottom line of using cardiomyocytes. Mainly to distinguish between HERG-only blockers or balanced ion channel blockers. For example, if it blocks in addition to HERG or calcium or HERG and lead sodium current, you can see that clearly in cardiomyocytes. Assay, ME assay.
Okay, thank you. Now, our next question, Carol, let's go back to you. How does the camera work? Does it take an image record of the whole plate or specific wells?
So the camera is focused on the bottom of the wells in all of the plate, and that would be a 90. It can be a 96 or a 384 or a 1536 plate. We have a technology that will find the center of the plate and record from that section for all of those wells in each plate at the same time. So you can crunch through a lot of data quickly.
Thank you. Renji, when are the new protocols expected to go into effect with FDA or EMA?
I think they are in the final stages. Having said that, they keep pushing the deadline or the guidelines every year. So I think it's going to be this year, 2020. But it all depends on all the consortiums have to come together and agree on it. Now, it is not a simple process of a few people agree together and say, yes, let's go with this. This is a global process. It's not only FDA, the European guidance, and the Japanese guidelines and the pharma industry. Everybody has to come together and agree to it. So I cannot say a clear answer, but it is coming.
Thanks, Renji. Now, Oksana, what is your experience with CDL iCell Cardiomyocytes 2 as compared with the old product Cardiomyocytes? Do you see fast appearance of spontaneous beating in Cardiomyocytes 2?
Yes, so actually, if you work with Cardiomyocytes 2, you can see the beating the next day, definitely the day after. So they get mature pretty fast and they're much easier to work with. So when I had a lot of experience with working with older version of cells, and this kind of requires quite prolonged time of feeding and culture before experiment, and with Cardiomyocytes 2, you can start your experiment much faster.
Thank you. Renji, in rolling out new drugs, do you see the need to get cardiomyocytes from each patient during clinical trials?
I wouldn't go that far, but there is some proposals going on. But basically, what we are looking at the cardiac safety is that if it goes out into the open population, healthy population, or people who are taking drugs specifically, will adversely affect them or not. So just looking for specific cardiomyocyte from a specific disease background, it is too far at this stage, but it may be coming. I don't know whether I'm having a clear answer to you, but it is a very far-fetched question.
Thank you. Now, Carol, has any work been done on modeling various peak characteristics to predict which ion channel is being affected?
So I think that's a really good question. I think we are just getting started with this, and I think the field is as well. So I think that's something that we can look forward to seeing in the future.
Thank you, Carol. Now, Oksana, on your flipper assays, how do you correct for the different cell density between wells?
So typically, the cell density is pretty consistent. At least we like it to be that way. So because the density is consistent, we have pretty consistent beating rate and amplitude. Usually, if you have variation with cell density, you would see big changes in amplitude and sometimes beating rate, or you may not have beating that well at all. So in these cases, we just exclude those wells, but typically we don't have to do that. And we don't have ways of normalizing for different cell density right now.
Thank you, Oksana. Renji, it was mentioned that you don't have to do the thorough QT assay if the drug passes through CiPA. Could you explain that more in detail?
Yes. So the CiPA, the 4 buckets, you look at the ion channels, and then based on that data, you do the modeling, in silico modeling, to have a pro-arrhythmia risk. That comes from the 28 compounds from the low, medium, and high throughput block. So if your compound, goes through the ion channel, all the specified ion channel in the first bucket, and then you model that, and if you have a pro-arrhythmic score, which is, say, it's a low risk, then, you have to test for this in the MEA to make sure there is any unwanted effects. And then you also have to do the phase 1 QT interval where, again, you want to make sure that there is no surprises for you in that state. If all of this comes clear, then you don't have to do a thorough QT assay in phase 3. And people who have done this, it's very expensive and very time-consuming, and that's one of the strengths of the CiPA initiative people are trying to achieve.
Thank you. Now we've had some great questions coming in. We have time for just a few more. Let's go with this one. Will CiPA replace need for careful clinical assessment of electrophysiologic effects in phase 1 ECG safety studies?
No, that's not the case. I assume this is for me, but if you look at the 4 buckets in one of the CiPA initiative slide, the last one, the 4th bucket is the phase 1 ECG. You still have to do it, but you are not looking at the QT prolongation. There are some new parameters they're trying to extract from this QT the QT parameters for specifically, for example, J to T peak and T peak to T end. These 2 peaks differ, in their response. At least, in a few positive controls they have tested, that the balanced ion channel block, does not prolong the J peak to T peak, whereas the HERG block only prolongs the T peak to T end. So they are trying to distinguish in these phase 1 studies pure HERG blockers or multiple ion channel blockers. Obviously, you will need balanced ion channel block in your drug to go further and to have adverse labeling, not to have adverse labeling. So yes, in a side answer, yes, you need to do the phase 1, but it's not going to be the same as what was done before. They are adding some new parameters to distinguish between the good and bad drugs.
Thank you, Renji. And let's wrap with this final question. Carol, I believe this is for you. How do you know what settings to use?
So as we've been doing assay development and through the validation process on our new camera and the Penta instrument, we have documented the settings that we used, and we've actually placed them in an 80-page assay protocol guide for different assays and things that comes with the ScreenWorks software to give people a good start at the settings that we've used, and then they can do further assay development from there.
So I just wanted to add a little bit. So it's actually pretty fortunate that exact setting is not even that sensitive for the quality of assay. So you can use a range of settings and your assay would be still kind of consistent and reproducible. So we propose some average settings, but they could be modified without effect on quality of experiment.
Thank you. Now, a big thank you to our speakers, Dr. Renji Ranganathan, Dr. Panita Larkiatmangal, Dr. Oksana Sharenko, and Carol Crittenden for their time today and their important research. We would also like to thank LabRoots and our sponsors, Molecular Devices and Eurofins Discovery, for underwriting today's educational webcast. Before we go, I do want to thank our audience for joining us today and for their interesting questions. As a reminder, the questions we did not have time for today and those that are submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we thank you for joining us. Until next time, Bye-bye.